Hello and welcome to Inside the Alzheimer's Lab, brought to you by Cure Alzheimer's Fund. I'm David Shank, author of The Forgetting and senior advisor to Cure Alzheimer's Fund, a not-for-profit scientific consortium dedicated to ending Alzheimer's as soon as possible. We bring you a series of discussions throughout the year to help improve your understanding of this disease and the science behind it. Today we're going to be talking with Dr. David Holtzman, who is indisputably one of the world's leading researchers on Alzheimer's disease. To summarize all of the things he's contributed would take the hour just to summarize it. We're going to be talking most specifically today about his contributions to a really interesting and cutting-edge aspect of Alzheimer's disease research, which has to do with the relationship of sleep to Alzheimer's disease. And I've noticed as the bulletins have gone out to people on our newsletter, that has gotten people's interest. I know this is something that's interesting to me and my wife and anyone who doesn't think they're getting enough sleep. So I think this should be a really interesting program. And we'll talk to Dr. Holtzman about some of the other really important research he's done. We'll also be taking your comments and questions as time permits. So what we need you to do there is click on the questions link below this screen, or you can go to a separate website, which is yorn.com. That's yorn.com/curealz, and you'll see directions about how to ask us a question. So let's talk to David Holtzman. Let me welcome him formally. Dr. Holtzman is the Andrew B. and Gretchen P. Jones Professor of Neurology and head of the Department of Neurology at Washington University in St. Louis. He's also the Charlotte and Paul Hageman Professor of Neurology and Molecular Biology and Pharmacology and Associate Director of the Alzheimer's Disease Research Center and a member of the Hope Center for Neurological Disorders. He's won practically every award you can get in this field. He's quite distinguished, as I mentioned. And most importantly, he is, for nearly the entire history of Cure Alzheimer's Fund, been a vital member of our research consortium. Dr. Holtzman, it's great to see you again. Thank you so much for joining us today.
Well, thanks so much for inviting me, David. It's a real treat to be on, and I look forward to talking about some of the work on sleep.
So I want to do two things before we get into some of the detail about your latest research on the relationship between sleep and Alzheimer's disease research. The first is we're going to tease a little bit of breaking news here because, as you just told me, a minute before we went on air, there's something very exciting happening in Washington today. We talk all the time about how we don't get enough funding for public funding for Alzheimer's disease research, and that's still going to be true after today, but it's going to be a little bit less true. It's going to be a little bit less bad, apparently. Why don't you tell us what you know and what the picture looks like?
Well, the spending bill before Congress is supposedly going to be signed today, both by Congress and the President, which would not only deal with spending for all of the things that we spend in the United States, but it would probably raise funding for Alzheimer's disease by about $350 million a year, which is obviously a big number. It would increase the current funding from about $500 million to around $850 million. So that's really a major change once that bill is signed. It will really stimulate the field, and we need it.
So we're looking forward to confirming that, and please don't take this as confirmation, but it looks close enough that we could at least
looks good. I mean, we'll wait till the final word, but it looks pretty good from what I hear.
And to put it in context, the figure that we throw around in terms of what we in the research community think we should be spending, federal dollars wise, is closer to about $2 billion. That kind of would raise it up on the level of how we treat other diseases of this significance. Is that right?
It would certainly, so it would certainly get it closer to some of the other major diseases like, say, cancer and heart disease.
Right. So that would be truly a great step forward, and hopefully we'll be announcing more news about that on our website, and people will be hearing about it in the general news that they follow. So that's the first thing I wanted to get that out of the way. It's really exciting. Puts me in a good mood. The second thing I want to do is talk about just generally about all the exciting things that you've done in Alzheimer's disease research, and we'll try to make this brief so we save a lot of time to talk about sleep. But people need to know that that you're one of those precious few researchers who, even though you look very young and you are rather young in the scheme of things, you've been around for a while and you've really touched on maybe every major aspect of Alzheimer's disease research in your time, which is a little unusual. You've been on the tau side, the tangle side, you've been on the plaque side. So could you just kind of give us a summary of how you got into the field, what the first thing you did was, and kind of the major touchstones of your Alzheimer's research career?
Right. Well, the first thing in my research career that really I started to work on that was directly relevant to Alzheimer's disease was actually during my postdoctoral training, when I began to do some work on animal models of Down syndrome, and that was because of the connection between the fact that everybody who has Down syndrome will develop dementia from Alzheimer's disease at a relatively young age, and so that sort of started some of the work that's more directly related to Alzheimer's disease. When I moved to Washington University and really started my own lab, the major projects that I started work on were on ApoE. So ApoE is the most important genetic risk factor for Alzheimer's disease, and there's one type of ApoE called ApoE4 that puts you at significantly higher risk for the disease, and if you have another form called ApoE2, it puts you at lower risk. And to summarize a lot of the work that we've done, it strongly suggests that the reason that these different forms of ApoE that we carry either are protective or risk factors is that they alter the buildup of the amyloid protein in the brain. So amyloid is a key protein that's involved in the initiation phase of the disease in the brain. And ApoE, if you have the bad form, let's say the ApoE4 form, it causes that protein to build up earlier and to a greater extent. And if you have the protective form, it actually has the opposite effect. It delays that process. And our lab was sort of showed that really in animals for the first time. And then we've subsequently worked a lot more on the details of exactly how it does that. So that's one major thing we've done. We also, when it first began to be realized that you could immunize people or animals with the amyloid beta peptide, and that the people or animals would generate antibodies against this protein, and that could potentially be a treatment. We were one of the first labs to demonstrate that using monoclonal antibodies, which are directed specifically against the amyloid protein, and one of the antibodies that we worked on early on had a name called M266, and we showed in animals that this antibody could reduce Alzheimer's pathology, it could improve the behavior of animals, and that antibody was further developed by Eli Lilly, and that became the antibody that's being tested extensively now in humans called solanezumab. So that antibody is in several phase 3 trials to see if it is useful as a treatment. The other work that we've done more recently is, we'll talk about sleep later, but about 6-7 years ago, we started to work for the first time on tau, which wasn't something I'd worked on early in my career. And through a series of experiments with my lab and also the lab of Mark Diamond, my lab developed several antibodies against tau, and initially as reagents, but then we were able to show that these antibodies were able to decrease the buildup of tau pathology in the brain of animals, and some of this work is now has been moving into human trials over the last 2 years. Those are some of the things that... Actually, David, I can't hear you now.
Thank you for telling that. So I had muted myself. Thank you for.
Okay.
So there's a lot there, and there's a lot that hasn't been said, and I'll just throw 2 things on top of that, which is that I forgot to mention that you're also an MD in addition to a PhD, which is not not unheard of, but somewhat rare in this world, and it gives. We've talked off air a lot about how that gives you added perspective to all of your research. And the other observation I'd like to make, just to share with our viewers, because I have had the honor of being in several research consortium meetings now, and seen you interact with specialists from all over the world, and I know firsthand that your breadth, your experience with all these different aspects of the disease, not just being a specialist in one of them, gives you not only does it bring a lot of respect for you to the room, so people want to hear what you have to say, but it gives you, I don't want to say unique ability, but a somewhat uncommon ability to chime in on an awful lot of different subjects and do a lot of cross-pollination and kind of connecting one thread to another, which actually is what Cure Alzheimer's Fund is all about. That's the whole point of having this research consortium. And you and I have talked several times about how special that collaboration is, and I'm not surprised that you feel that way, because you're really able to take special advantage of that collaborative nature.
Well, thanks. I've been fortunate to be in a situation where, while I spend a fair amount of time doing, having a lab that does basic research, I have the connection both of seeing patients, but one of the other things I wanted to mention that I've been involved with over now, gosh, 15, 16 years is also trying to understand where some of these molecular markers of the disease that we study all the time in animals as well as in people might serve as biomarkers for the disease. And when we talk more in a moment, I'll get into that. That's actually been a really good way to connect some of the basic science to human disease more directly.
All right, so the viewers, the non-scientist viewers, do not have to remember all those different details, but I want them to have a sense of the overall picture of your tremendous breadth and how that enhances all the particular things that you do. So now let's talk about sleep. You brought some slides, and you're going to kind of run us through a reasonably short and lay-friendly presentation, I hope, and then I'll ask you some deconstructing questions when you're done. If we're ready to pop those slides up, let's begin.
Okay, so I think one of the important things that I think about all the time that's been discovered over the last 15 years or so is that if somebody develops a dementia that is from Alzheimer's disease and begins to develop memory loss and other cognitive problems, but is in the very mild stage of the disease clinically, so let's say they just become forgetful but they're otherwise able to still do a lot of their normal activities, what we often term that stage of the disease is a very mild Alzheimer's disease or mild cognitive impairment. And many people who have that stage, if the underlying cause for their cognitive impairment is due to Alzheimer's disease, and if they happen to die for some reason, and you look under the microscope at their brain, or by using different markers of the disease in living people, such as things like what we call amyloid imaging, or we do cerebrospinal fluid assessment to measure whether Alzheimer changes are occurring, the very mild clinical stage of the disease is not the beginning of the disease. If you look on this slide, in the red line shows a representation of the amount of what's called amyloid that's building up in parts of the brain over the course of someone who has Alzheimer's disease. And what it shows is that by the time you're very mildly impaired, there's already a lot of amyloid buildup in the brain. This protein that we all make, but it normally doesn't clump together and build up. And the other major pathological feature of the disease, the buildup of what's called neurofibrillary tangles, even at the very mild stage of disease, is not at its peak, but it's already on its way up. And so what this shows really is that in most people that are developing, let's say, they have pure Alzheimer's disease as a cause for their cognitive decline later on, that pathological process, if you see the red line, amyloid plaques, probably started about 20 years prior to when they became cognitively impaired. I think that's the good news about this is that, one, that means if we can identify this stage of what we call preclinical Alzheimer's disease, when people are developing these changes in the brain but they're still normal, that might be a time to really maximally intervene, or even before that. And the other thing that this graph suggests is that if we can figure out ways to block this pathology, either with drug treatments or even lifestyle factors, that might delay the onset of disease much later. And so some of the things that we really need to do in the field in this preclinical stage of disease, when you're developing this Alzheimer change but you're still normal, one of the things we can't now tell, other than using these biomarkers, is whether individual people's function is abnormal in some other way that we can't detect yet. And that I want to talk about that more in relation to sleep. So I think we can go to the next slide. So this is might be a little complicated to understand, and I'll just go through one or 2 points on this slide. There's a lot of evidence in the field that all of us make this protein called amyloid beta, or Abeta, which is shown on the left side of the slide. And it normally is produced by our brain, and it's cleared away within hours, and it doesn't generally build up. However, for a variety of reasons, genetic factors, aging, and other things we'll talk about, in some people it will clump up. And when it clumps up, it forms these things called aggregates, Abeta aggregates, or amyloid. And those aggregates in some way seem to trigger the Alzheimer's disease later on by both directly damaging the brain, instigating inflammation in the brain, but also causing this other protein called tau to also build up in the brain. And so some of the things that I'll talk about next are there things that can influence this process other than, let's say, drugs, for example, that are relevant for either diagnosing the disease or even affecting its or treating it. So if you look in the middle of the slide, one of the things I'm going to tell you next is that the sleep-wake cycle, or how much we're awake and how much we're asleep, actually directly affects the level of the amyloid beta peptide in the brain. And by affecting that level, it looks like it can influence whether or not this amyloid beta peptide aggregates or builds up in the brain. And I think that's important because it suggests that the sleep-wake cycle itself is an important factor that might be related to the beginnings of Alzheimer's disease and even the symptoms, as I'll talk about in a moment. So if you go to the next slide. So I just wanted to go through historically how I even got interested in this briefly. So one of the graduate students in my laboratory now 6-7 years ago, Jae-Eun Kung, was working on a project, and we had developed a methodology to measure this amyloid beta peptide in the brain of living animals. These are mice. And these animals, you can see on the graph at the top that what we're showing is the levels of the amyloid beta peptide every hour over a 72-hour period. And what we found, and just sort of stumbled across this finding, is that the amyloid beta peptide is higher during the 12-hour dark period and lower during the 12-hour light period. And if you remember, rodents or mice are nocturnal. So during the dark, they're more awake, and during the light is when they sleep more. So this finding immediately suggested that perhaps somehow the sleep-wake cycle normally regulates the levels of this peptide in the brain. And some of the other experiments I won't go through here showed that if you sleep deprive an animal, the levels go higher, and if you cause them to sleep more, the levels go lower. So if you go to the next slide, some of the subsequent experiments that we've published were that if you have these animals develop amyloid deposition in the brain, which is shown on the right. All those brown dots are the buildup of this amyloid protein in the brain of these genetically modified mice. And when we sleep deprive the animals for, in this case, about a month and a half, they developed a lot more amyloid plaques in the brain, like 2.5 times more in this relatively short period of time. Now you have to remember, if you try to equate this to a human, it's not the same time frame. That these kind of changes over months are more equivalent to years. But in either case, it was a very profound effect. And the other thing we found, which isn't shown here, is that if we cause the animals to sleep more by giving them a pharmacological treatment, that actually lowered the amount of amyloid deposition. So if you go to the next slide. So that's all of those findings suggested that if you modify sleep, that modifies the formation of Alzheimer's pathology, at least the amyloid part of the pathology. So one of the next series of things we wanted to ask is, once amyloid starts accumulating in the brain, does that itself disrupt sleep? So if you look on the bottom panel here, what it's showing is the amount of minutes awake per hour in mice as they build up amyloid in the brain. So at 9 months of age, these animals have a lot of amyloid, and if you look under in the bar, the amount of minutes awake during the light phase is about 30 minutes per hour, and that's about 50% more. They're awake about 50% more than normal as they develop amyloid deposition, and that's shown better on the next slide. So in the next slide, we'll see evidence that was data from Ji-Hoon Roh, an MD, PhD, who was in my lab that studied these animals. And if you look in the middle panel, G, what you can see is during the light phase, when the animals should be sleeping they should only be awake about 20 minutes per hour. By 9 months of age, where it says 9M, they're awake 30 minutes per hour. So that's a significant change. So they're basically developing abnormalities they're awake too much as they develop amyloid deposition. So if you go to the next slide, we wanted to ask a question: if you prevented the accumulation of amyloid in these mice, would that normalize their sleep? So if you look at what we did, is a well-known now procedure in the field where we actually immunized the animals. We injected them under the skin every month with the amyloid beta peptide itself. This would be analogous to immunizing you against the flu, except in this case to prevent you from getting the flu. But in this case, we're using this protein to immunize the animals. The animals then develop antibodies to this protein. And if you look in the top two images, there's no brown spots in the brain because those animals that were immunized didn't develop the amyloid plaques, whereas the animals that weren't immunized or immunized with a control have a lot of amyloid plaques. So then we analyzed these animals before we looked at their brain, and if you look on the next slide, what you will see, if I have you focus once it appears on some of the panels, we looked at their wakefulness, and we found that the animals that were not vaccinated had very abnormal sleep, like we had shown before, but the animals that were vaccinated, their sleep was normalized. So I don't see that slide, but I'm just explaining what we have found. So I think these studies in general suggest a model whereby not enough sleep or too much wakefulness, which could be due to a variety of factors. It could be due to sleep-disrupted breathing or sleep apnea, which is a very common phenomenon, or any reason why people develop sleep problems. If that happens in midlife for periods of time, it might suggest the possibility that that would accelerate the process of developing amyloid deposition. And then once you begin to develop that amyloid deposition in the brain, that further, in a feedback loop, might cause more sleep problems. So that's the kind of thing that we and many others around the world are beginning to test to better understand this whole process and how Alzheimer's changes might be accelerated by abnormal sleep, and how if we potentially optimize the amount of sleep we get, we might be able to decrease the risk of Alzheimer's disease. So we can talk more about this, but that's kind of where we're going now. Our group has published some work in humans where we've studied individuals who are late middle age, some of whom have amyloid deposition and some don't, and we find that they're sleep efficient if they have amyloid deposition in the brain. Even when they're still cognitively normal, their sleep efficiency is not as good. And that would suggest, like we found in animals, that somehow the amyloid buildup is actually causing sleep problems. So this is leading us now to do a whole series of further studies where we're very carefully studying sleep at home in individuals with simple devices to really determine whether sleep abnormalities might be an early biomarker for the disease that we could then use to determine if treatments would influence that process. So that's some of the things we're doing now. We can talk about a lot of other things. But
did we get to all your slides, David, or
the last slide wasn't shown, which is a kind of the hypothesis slide of how we're thinking about this.
Okay, is that this one here?
It is up now, correct.
Okay, so is there anything you want to go over there, or do you feel like you already covered that?
I pretty much covered it. What's not on what I didn't discuss that's related to this slide is there's a variety of other changes that we're not looking at in this summary slide, which is that some of the other factors that influence Alzheimer's disease, like tauopathy or neurofibrillary tangles or neuroinflammation, those might also play into this process, and that is something that could be further addressed in the future. But I wanted this is at least going over how we think about how sleep might influence the amyloid beta protein and how amyloid deposition itself might feed back on causing sleep problems.
Okay, so really interesting stuff. We have a number of questions pouring in as we expected, because this is such a topic that just obviously touches people's lives and imaginations and also fears. So I think the first question is, how do you straighten out cause and effect? Someone asked about, a viewer asked about this. How do we know that we're not just seeing the beginnings of Alzheimer's disease causing sleep problems, and it's kind of the Alzheimer's is causing the whole cycle first, as opposed to the sleep really contributing? You may have already said that, but if you could.
Well, I think it would take a long time to address that in human beings, but at least in animals we can actually direct. That's one of the reasons I showed the slide in animals, where if we have animals that are going to develop amyloid deposition, and we simply prevent the amyloid deposition from building up, that prevents the sleep problem. So at least in that relatively straightforward model, that argues very strongly that the amyloid buildup is causing the sleep problem. Now, in humans, in order to prove the same thing, you would need to do an experiment that would take quite a long period of time to know that. In other words, if you had a treatment that prevented in humans the buildup of amyloid, and you showed that prevented sleep abnormalities versus in people that weren't treated, I mean, that would get at it as well. But that would take a long time to prove.
There was a question about the different types of sleep and how we would know if we as humans are getting the right kind of sleep and are in the proper deep sleep, and also what the relationship of the different periods of sleep might be to the research you've done. Anything there?
Well, this is still now what I'll say is speculative, but what we think is that, at least in regard to what I've been talking about, how sleep might interact with the amyloid protein, the reason we think that really good deep sleep, or what's called non-REM sleep, is what we want to optimize, is that when you have stage 2 and 3, what's called non-REM sleep, your synaptic activity or neuronal activity in the brain really drops to low levels, and that is synaptic activity or neuronal activity is what drives the levels of the amyloid beta peptide higher. So at least in regard to that hypothesis, we really think that deep sleep is really what's beneficial. Now, there's a lot of other studies that are not related to Alzheimer's disease that also show that this deep sleep is very beneficial as well for memory, keeping your brain renewed, etc. So it's not just for this Alzheimer process, it's probably a good thing to get deep sleep, but also for just normal brain function.
And is there a number in your head of what I should be, the number of hours I should be sleeping tonight?
What's interesting is that if you study normal people, well, one, during aging, everybody's sleep, the amount of time they sleep goes down. That just happens normally. But everybody, if you take people at different age ranges, the amount of sleep people have is quite distributed, even if they don't have a sleep problem. So it could be anywhere from, let's say, 5 hours to 9 hours a night. And you see a normal distribution, even in people that are okay. So there probably is an optimal amount for each person. That's not necessarily, it's hard to pin down a number, but if you threw something on, most people say like 7 to 8 hours, but it's really variable depending on the person, what's optimal.
And is this something, I know that we're stepping outside of your research, but I'm sure you're curious about this and know more about it than I do, certainly. Is this something that that we should know, just kind of knowing ourselves, we get up in the morning, we feel rested, we feel like we've gotten enough sleep, that's okay? Or is this something we actually should investigate somehow to see if we're actually getting enough for our own?
Yeah, I think the issue from a medical standpoint, and not even necessarily related to Alzheimer's use, just if you have, when is it that you should be worried that you have a sleep problem, for example? I mean, I think if you if you're waking up in the middle of the night, that's not normal. You shouldn't do that a lot. If you're when you wake up, if you're not feeling refreshed, that happens with normal sleep. So if you have a partner and they're saying you're snoring at night all the time, that might indicate you have sleep disordered breathing. Those are all things really you want to go see a physician about, because there are things that might indicate you have a sleep problem that really needs to be addressed and treated. And if you really you feel like you're sleeping well, you feel refreshed when you wake up, and you don't necessarily have to be alert, be worried about that. You'd only really get worried if you show symptoms that might relate to a sleep problem.
Because I think one common experience in our modern world is that people don't get enough sleep, or they're going to bed late because they have stuff to do in their lives. They're getting up early because they've work to go to. They don't feel great when they wake up, but they wake themselves up somehow, maybe with exercise or with a cup of coffee or 5 cups of coffee, a few more cups of coffee, through the day, so they don't feel the tiredness through the day. They kind of wake themselves up in various ways, and then they get, they go to bed, they crash, and they have some sleep, and they start all over again, and they're not really sure if they've got a sleep problem because they don't feel super rested, but they are using caffeine and some other methods to at least give them the energy they need during the key hours of the day.
Right. So those kind of, we just speculating. We think that kind of behavior probably isn't good for your brain for all the reasons, many of the reasons I just talked about.
Right.
So one of the things we know, there are certain things that optimize sleep. So regular exercise optimizes sleep. Not drinking or intaking caffeine right before you go to sleep. Not drinking too much alcohol before you go to sleep. Those things actually do disrupt sleep. So those are just common sense things that we do know can make a big difference.
Right. And we know that there's a connection between exercise, a preventative connection between exercise and Alzheimer's. So these things are obviously...
And it's possible that part of that is because not only the exercise direct benefit, whatever that is, but it may be that exercise by optimizing your sleep also is how exercise helps.
Wow, that's interesting. Is anyone doing that particular study to know kind of where the cause and effect?
I'm not aware of anybody's actually looked at that directly to see how that interaction, how one is affecting the other, and then is that do you get the benefit because of that? I'm not sure, but it certainly is known that it will.
All right, maybe I'll take that to the next RC meeting and suggest it to Rudy and see if someone wants to take that up. I'm sure I'd get some good respectful looks there. Speaking up when I'm not supposed to, when I'm just supposed to be listening to the scientists. Boy, there's just so many questions to ask here. Is there any evidence? This is from a viewer, that if you built up the amyloid due to lack of sleep, and then you start getting more sleep. Yes, I was wondering this also. Yeah. Will that reduce the amyloid levels? Will it possibly reduce the plaques? That is the kind of more permanent buildup of the amyloid. What do we know about that so far?
Yeah, we don't know the answer to that at this point, but I would think that there's a good likelihood that it would, if you normalize your sleep once you had the amyloid, it would probably prevent the growth from being as fast, because basically the production of the protein would be more optimal, so it wouldn't be as high. Mm-hmm. Or also the clearance of it. Based on other work from groups like Macon Niedergaard's lab, the clearance of the protein might be improved as well. So I suspect that it would slow the growth, which is a good thing. But again, that's not been directly assessed in it.
But it's kind of hard to imagine that you would be able to degrade the plaques once they're formed. Is that
I think, well, the problem is that there are ways to degrade the plaques, but usually the things that have been proven that do that are more interventions that are biological treatments, or drugs, things like that, or right. Antibodies. But once that protein forms these clumps, you can, if you block the production or enhance the clearance of what's called the soluble amyloid protein, that will prevent the further accumulation, which is a good thing, but it generally doesn't remove these clumps.
I think we should also say, and please correct me if my understanding of this is not right, but I think we should also say that we're really, all of this stuff, sleep, how it affects amyloid, how it would then affect the onset of the disease, is really all kind of early talk, the early stages of the disease, because we get to a stage of the disease where once the amyloid has then caused the tau to form, the tangles that we've talked about, that people know about, and then subsequent to that, inflammation in the brain, there are new cycles going on, right, which the amyloid deposition aren't necessarily going to be able to, or the lowering of amyloid aren't necessarily going to be able to.
Once I think, if you related this to humans, once human beings develop cognitive impairment from Alzheimer's type changes, it might be that affecting amyloid would have some effect, but it seems to me unlikely that it's going to have nearly as big of an effect as affecting these other processes at that level, whether it's inflammation, tangle formation, etc. So I think one of the other things that we haven't talked about is that it's been known for a long time that as people develop dementia from Alzheimer's, it's very frequent that their sleep is can become quite disturbed. That's been known. But we also haven't touched on the fact that one of the things that's important in regulating sleep is the circadian rhythm. So that's the circadian rhythm, which is what gets thrown off when we have jet lag, is also very relevant for sleep, and they impact each other. So as people develop clinically Alzheimer's disease, have cognitive impairment and get demented, their circadian rhythm also becomes disrupted. So there's sort of two things probably going on at the same time. There's sleep disruption per se, but there's also the brain's clock becomes disrupted.
And that's really more later stages of the disease, not really connected to the specific research you've been talking about.
As far as what my research has looked at, we haven't in this sort of early phase of the disease where you're getting amyloid deposition and you're still cognitively normal. We don't have evidence yet that the circadian rhythm is disrupted. But one of my close colleagues who was in my lab, he's now an independent investigator, Eric Music, here at WashU, is actually he's doing really interesting studies looking at how the clock genes, our body has these genes called clock genes that regulate our, daily rhythms. And those clock genes do appear to play a role in affecting neurodegeneration. And some of those mechanisms may be overlapping with the sleep mechanisms, and some may be completely distinct. So that's something that's really in its infancy and just starting to be studied now, the relationship between the clock or rhythm and Alzheimer's disease.
All right, I've got a good curveball for you. This is a fantastic question. I never would have thought of this, but I I don't know how we'd research this in mice, but what about the possibility of essentially substituting meditation for sleep? Is there anything that we know about what meditation does to the brain?
Right.
At least to the human brain. I don't know if we've been able to see mice meditate, but that would make up for possibly some of the damage that you're apparently getting from not getting enough sleep.
No, I think this is a great question. The sort of idea of mindfulness and meditation, and could that improve health,
Right.
including this kind of thing. I think it might be a little hard to understand how to do a study like that in mice where you can't communicate or instruct an animal like that to do
Right.
that. But I think what's starting to be approached in humans is studies where people meditate, and then they're studied in like an MRI scan that can look at the brain's activity during that. And I'm not aware what those findings. I think there's been some work on that. I can't remember off the top of my head what the findings are. But if it ended up showing something that you could then mimic in animals, then maybe you could. In other words, let's say the brain's activity changed in a certain way during meditation. You could try to then mimic that same change in an animal and see if you got beneficial effects.
Right, right. So is there going to be a way to take this particular research to human beings? Where do we go from here in terms of what you've already started?
There's lots of things I think one can do in humans to test this. I'll just give you a flavor for some of the things we are doing. I'm sure there's many more that you could do. So I've always, many of us here at my university have been very interested in this sort of preclinical phase of Alzheimer's disease, because we think that would be a way to, if you could treat people at that point, you could then delay the disease from manifesting. So that's, I think, really important. For example, there's a large study that Risa Sperling and Paul Azen and many people around the world are involved with, called the A4 study. And that's where you identify people who have amyloid deposition in the brain that are cognitively normal, and they're putting them on a randomized treatment to see if that will delay the disease. So one of the things that the field really needs are functional tests that you can do in this preclinical phase of disease, because people are cognitively still okay. How do you know that their function is in any way impaired? And what we're trying to understand right now in a study we're doing in humans is whether, if we monitor sleep in people in this phase of disease versus in people that are cognitively normal that don't have the buildup of amyloid, are there abnormalities in sleep that we can pick up using simple at-home tests? So let's say, for example, let's hypothesize that non-REM or deep sleep becomes impaired during that phase. We can determine that by doing these home sleep studies. And if that's true, we could then monitor people in these treatment trials to see, maybe earlier than waiting 5 years, if their sleep was getting better. And that's not now. This is just an experiment we're doing. I mean, if it really works, it would suggest a new marker for, is a therapy working that doesn't rely on, let's say, subjective reports. Measuring sleep is very quantitative. You can get real numbers exactly, not only how long people sleep, how deep they sleep, how strong the deep sleep is. All of these things can be accurately measured with very simple at-home tests. So that's what we're trying to explore now in humans, to determine whether we can pick an abnormality up or not in that phase of the disease.
If I could ask the producer to bring us back to the very first slide, I wanted to ask you a question about that connects to what you were just saying, because this is a point we really, I think, need to go over and over again in not just this discussion, but subsequent discussions about what we're finding in the research. There's a very long period where you don't really see any symptoms of the disease,
right?
And but we do know that the amyloid is building up.
Amyloid's building up, and even the tangles are starting to build up too later.
Right. So I don't know if we've got that slide up. You could probably tell me, David, if you're seeing that first slide.
I can see it on. Yes, I can.
Okay. So I don't want people to be misled by this. I feel like I want to clarify something. The line that says neuronal integrity, that's essentially cognitive compromise, right? I mean,
well, no, not necessarily cognitive compromise. It means that, so we have a lot of, fortunately, our brain has a lot of redundancy. So we think that during this phase of disease when the amyloid or the Alzheimer pathology is accumulating, that the nerve cells in the brain are beginning to not function as well, and some might be even dying. But because the brain has a lot of redundancy, there's no cognitive abnormality until later.
Right.
So you probably need to hit a threshold of abnormality where once you've lost enough cells or once you've lost enough connections or synapses, only then is when you begin to, maybe that threshold is losing 30% of your synapses or 40%. I don't know what that number is, but there's some point in which, then you begin to manifest symptoms.
So to look at this slide, I'm wondering, would we want to see a fourth line here, which is the cognitive impairment? And if we did, you probably know, you could probably tell us, where does that line, let's say there's a yellow line.
Yeah, on this slide, the cognitive impairment is shown on the bottom line. So in other words, preclinical AD, it says non-demented.
Okay, excuse me.
Clinical dementia rating 0. That's normal cognition.
Okay, so not until we get to where it says very mild AD.
That's where the cognition changes.
Okay, so that's really a point to just hammer home. I hope people can understand that. Everything, if you look at where the line begins where it says very mild AD, everything to the left of that is the disease happening, but no one, including the patient presumably, aware that there's any disease happening.
That's exactly right, and that's why this is so important, I think, as a concept. And not only is it an important concept for this disease, most of the other what we call neurodegenerative diseases probably have a very similar diagram you could draw, where let's say in Parkinson's disease or in amyotrophic lateral sclerosis, you're probably developing whatever the underlying pathology is for those diseases 10, 15 years before or 20 years before as well, and they accumulate, and then once you hit some threshold where that pathology is leading to enough damage in the brain, only then do you manifest symptoms.
So what Rudy was talking about at our recent symposium, and I think something that we really need to integrate into our conversations going forward, is to make an analogy to cancer, because I think we've been struggling with trying to communicate this idea, but we haven't quite hit it yet. People in the general in the world out there think of Alzheimer's as being associated with the symptoms, and we've been saying for years, well, the disease really begins before the symptoms, but we need to help people understand that that like cancer, where you can detect a cancerous tumor in your body before you are experiencing any symptoms from that tumor. Yeah, and you might have a tumor in your thyroid for years and years, or somewhere in your body, and actually not know anything about how that's hurting you, and it might not even be hurting you, except for that we've done a scan and we know that it's there, and they're trying to fight that the disease that's there, but we don't see the symptoms.
Right. I think something analogous, another medical issue that would be even perhaps more analogous would be atherosclerosis that causes heart attack, for example.
Right.
I mean, that starts occurring many years before, basically an artery starts getting clogged up, but you have no symptoms. It doesn't cause a problem until it gets critically clogged up enough, and then you have a heart attack or if it's in the brain, you get a stroke. But that process is something that, by understanding that, it was figured out that if you lower cholesterol with drugs, you could delay or prevent having a heart attack or a stroke. It's a very similar issue here. If we could delay, if we knew that was starting to happen, you could delay or prevent the accumulation of the Alzheimer pathology if you had the right lifestyle changes, treatments, etc.
Right. So it's so important for people to realize that the disease is beginning way before you see there's any inkling of any symptoms.
Absolutely.
And I think you said 20 minutes ago that, as best you can tell, that's really about 20 years before.
And that's also been very much substantiated in a unique population of individuals who have what are called dominantly inherited, a form of Alzheimer's called dominantly inherited Alzheimer's disease, where they have a mutation in a gene that if they have that mutation, they will get Alzheimer's disease. And depending on the mutation they have, it could start clinically where they get dementia, starting anywhere from age 25 to age 50, depending on the particular. And if you in studies that have been carried out in large cohorts of people across the world with those mutations, those people also start getting amyloid deposition about 20 years before they get symptoms. It's just like the late onset form. And then they start getting neurofibrillary tangles about 10 years before they get symptoms.
Oh, is that right? So the tangles also are coming in many years before you see the symptoms.
Absolutely.
So there's a great question connected to this, which is that now that we know this, I mean, it's frustrating. I just went to see my GP. There was no talk of checking my amyloid level. When are we going to be able to check our amyloid level so that we can know if the disease or the probable disease is actually already started in our bodies?
Well, so there are at least for amyloid deposition, there is an approved PET scan that one can obtain, but it's only approved by the FDA for by the government or Medicare. You can obtain this, but it's only for people who are symptomatic, and insurance will not pay for it, because if we know as of today, if we know the result, there's not a treatment that we could give that's different than if you didn't know the result. However, things are changing right now. One, if a treatment is ultimately approved that is a disease-modifying treatment, and you need to know whether or not somebody has amyloid deposition, for example, to give the treatment, then I'm sure it will become paid for by insurance and worth obtaining.
Well, this is a lay argument from a non-scientist, but it seems to me we already know enough about sleep and exercise and diet's connection to Alzheimer's disease, and so presumably connection to amyloid buildup, such that if we were able to get that early scan, and if it wasn't too invasive or hurtful, we'd all want that at a certain age so that we can take extra measures to modulate that buildup.
Right. I think generally I agree. The only thing is that right now, all of the things from a lifestyle perspective, whether it be sleep, exercise, etc., we would recommend that regardless of whether you're. So that's I think one of the why the government hasn't approved a payment for this. Now there is a big study that's starting later this year or later in 2016. Where I think it's Medicare is funding getting amyloid scans on 20,000 individuals across the United States to determine whether or not obtaining these will be medically useful, even without having another treatment. So this is actively being studied, will be studied. We obtain them now for research purposes in research studies, but not.
Right. Well, so hopefully we will get to that point where we're able to just routinely check our amyloid. I think that it seems like there's good cause for that. There's another great question about the amyloid vaccine and how that would probably play into what you're seeing with sleep. Is that right? If we can get a working vaccine that.
Right. So whether it's giving a single antibody to amyloid beta or a combination of them or doing active vaccination, certainly that slide that we showed would argue that if you're going to impact the disease the most, you'd want to start the treatment before symptoms start. And for the first time now, there are a few studies where that's occurring in this A4 trial that's a national NIH-sponsored trial in the United States. There's also in the dominantly inherited Alzheimer population, there's a DIAN study that is doing also pre-symptomatic treatment with antibodies. And then in the country of Colombia, there's a large population of people with dominantly inherited Alzheimer's disease, and they're also in a large trial. That's a secondary prevention trial where these antibodies are being given to see if they will delay the onset of dementia. So this is starting. It's not, but I think that's where the field is kind of moving.
Lots more important work to do. David, thank you so much. That is all the time we have for our program today. Thank you for watching. If you have any more questions about the science in today's show, please post them on our Facebook page at facebook.com/curealzheimers. Visit us regularly on that page. We post all sorts of research news and other news about the disease. As a reminder, this program and all of the webinar programs that we do are archived at our website, which is www.curealz.org, curealz.org, pretty easy to remember. Inside the Alzheimer's Lab, this program is a production of Cure Alzheimer's Fund, a not-for-profit dedicated to supporting the research that will stop this terrible disease. Every dollar you donate to Cure Alzheimer's Fund goes directly to scientific research. That's because our board of directors covers all the administrative costs. That means if you give $1 to Cure Alzheimer's Fund, that is $1 that is going to go into a lab. We're not going to take 39 cents of it and pay for administrative costs. Our generous board of directors covers all those office costs. So special thanks, of course, go to those very board members: Chairman Jeff Morby, Jackie Morby, Henry McCants, Phyllis Rappaport, Robert Greenhill, Matthew Zulik, Sherry Sharp, and our CEO and President, Tim Armour. And thanks again to our guest, Dr. David Holtzman. It's been great to talk to you, David, and can't wait to hear more about what's to come in your research. I'm David Shank. Thank you for watching. Have a good day.